The mirror test, a well-known measure of self-recognition, is successfully completed by mice. Greetings, everyone. Today's video focuses on the fascinating subject of mice and their ability to pass the mirror test, which is widely regarded as a reliable measure of self-recognition. In a recent study published on December 5th in the esteemed journal Neuron, researchers made a remarkable discovery. Mice exhibit behavior that closely resembles self-recognition when they encounter their own reflection. The groundbreaking research received support from prestigious organizations such as the Endowed Scholar Program, the Brain and Behavior Research Foundation, the Daiichi Sankyo Foundation of Life Science, and the Uehara Memorial Foundation. Now, without any further delay, let us dive into our engaging discussion on this captivating topic. In a recent publication in the journal Neuron, scientists have revealed that mice exhibit actions reminiscent of self-recognition upon encountering their own reflection. To test this, researchers applied a small white mark on the foreheads of black-furred mice and observed their behavior in front of a mirror. Interestingly, the mice dedicated more time to grooming their heads as if attempting to remove the ink spot. However, it is important to note that this self-recognition-like behavior was only observed in mice that were already familiar with mirrors, had interacted with other mice of similar appearance, and when the ink spot was of a noticeable size. By delving into the intricate workings of the hippocampus, the researchers were able to pinpoint a specific group of neurons responsible for the formation and retention of our visual self-perception. This groundbreaking discovery sheds light on the previously enigmatic realm of self-recognition, which had remained elusive in the field of neurobehavioral research. Neuroscientist and senior author Takashi Kitamura from the University of Texas Southwestern Medical Center explains that in order to create episodic memories of our daily experiences, our brains collect and retain information about various aspects such as location, objects, time, and individuals involved. The crucial element in this process is self-information or personal status. While researchers typically focus on studying how the brain processes and identifies others, the role of self-information remains unclear. To explore the mice's ability to perceive alterations in their own physicality, the scientists employed a mirror examination. Specifically, they applied a spot of ink to the foreheads of the black-furred mice, thus providing both a visual and tactile stimulus. While the mirror test was initially designed to assess consciousness across various species, the researchers emphasized that their findings merely demonstrate the mice's capacity to detect changes in their own appearance, without definitively establishing their level of self-awareness. Under specific circumstances, researchers discovered that mice have the ability to detect changes in their appearance. When mice were already familiar with mirrors, they exhibited a notable increase in grooming their heads in front of the mirror when marked with white ink patches measuring either 0.6 square centimeters or 2 square centimeters. However, no increase in head grooming was observed when the ink was black, matching the color of their fur, or when the ink mark was smaller, 0.2 square centimeters, even if it was white. Furthermore, mice that were not previously exposed to mirrors did not exhibit any increased head grooming regardless of the scenario. According to Jun Yokoze, the first author from the University of Texas Southwestern Medical Center, the mice had a distinct need for substantial external sensory cues in order to pass the mirror test. In order to achieve this, a copious amount of ink had to be applied to their heads, and somehow the tactile stimulus resulting from the ink enabled the mice to detect the presence of the ink on their heads through a mirror reflection. In contrast, chimpanzees and humans do not require any additional sensory stimulus to successfully complete the mirror test. Through the utilization of gene expression mapping, the scientists were able to pinpoint a specific group of neurons located in the ventral hippocampus that exhibited activity when the mice perceived their own reflection in the mirror. Upon selectively disabling these neurons, the mice ceased to engage in the grooming behavior triggered by the presence of the mirror and ink. Interestingly, a portion of these self-responsive neurons also became active when the mice observed other mice belonging to the same strain, characterized by similar physical features and fur color. However, this activation did not occur when the mice encountered a different strain of mouse with white fur. In order to determine the role of social experience in mirror self-recognition, the researchers conducted a study involving chimpanzees and mice. Previous studies with chimpanzees have indicated that social experience is necessary for mirror self-recognition. To test this hypothesis, socially isolated mice were included in the study. Surprisingly, these socially isolated mice did not exhibit increased head grooming behavior during the ink test. Similarly, black-furred mice that were raised alongside white-furred mice also did not display this behavior. Additionally, gene expression analysis revealed that socially isolated mice and black-furred mice reared by white-furred mice did not develop self-responding neuron activity in the hippocampus. This suggests that mice require social experiences with similar-looking mice to develop the neural circuits essential for self-recognition. According to Kitamura, there is a specific group of neurons that respond to self-stimulation. And interestingly, these same neurons were also activated when the mice were exposed to other individuals of the same strain. This finding aligns with previous research conducted on humans, which demonstrated that certain hippocampal cells not only fire when individuals observe themselves, but also when they observe familiar individuals such as a parent. The researchers have outlined their future endeavors, which involve unraveling the significance of visual and tactile stimuli. They aim to determine if mice possess the ability to detect alterations in their own reflection, even without any physical contact, potentially employing technology reminiscent of the filters found on popular social media platforms that enable users to adorn themselves with puppy dog features or bunny ears. Additionally, they intend to explore additional brain areas that could potentially play a role in self-recognition, as well as investigate the mechanisms of communication and information integration between these various regions. Yokosi explains that with the newly acquired mouse model, they now have the ability to modify or observe neural activity in order to thoroughly examine the neural circuitry responsible for triggering self-recognition-like behavior in mice. The Endowed Scholar Program, the Brain and Behavior Research Foundation, the Daiichi Sankyo Foundation of Life Science, and the Uehara Memorial Foundation provided generous support for this research. To support our channel's growth and ensure broader awareness, kindly hit the like and subscribe buttons. This will help us reach more individuals and disseminate valuable information. Thank you in advance.